So I was saying, I'd like to meet with people if possible this week about their projects, even if it's just half an hour. Help people get moving. If people could start out, I mean, people could just give a verbal update about how they're doing before we get started.
Or any problems that they're running into.
But let's just go around the room and have everybody. I mean, if the answer is you haven't done anything, that's okay too. But let's just hear from everybody. Antonio, how are things going?
I have not made much more progress. I've taken some notes on some of the recent articles you've given us for homework because in modeling TB, where I want to include kind of the mutation rate as well as antibiotic resistance, over the last two homeworks, we've kind of been dealing with a lot of that. So I've just been kind of taking notes and seeing how I can incorporate that into my model as well.
Right, because both that homework problem 2 on HIV and then the one in class also are both resistant strain competition models.
Right. Yep.
But we should meet on that. Kevin?
Yeah. Can you hear me? All right. So since the last time when me, you, and Joshua met to discuss it, I kind of made slow work on plugging in different equations for the virus spread term and the cancer growth term. So working those out with parameter scans and just see what's important out of those. And so yeah, I haven't made a lot of progress, but I've been working through those slowly. And that's all for that.
Okay. Let's keep. Let's see. Sorry, who's next on the screen? I'm sorry, I lost my screen here for a second. So, whoever's next, please go ahead.
I can go ahead and go. Can you hear me?
Yep.
So I pretty much just reworked some of my equations because I think I had them out of order, and yeah, that's pretty much it. I haven't done a whole lot of work, honestly, but basically just straighten it up and trying to get the work on the next step.
Again, I think if we make even half-hour appointments on a weekly basis, then that motivates you to spend a little more time on it. So it's probably not a bad thing to do that. And one-on-one we can work on it. Brian?
Hi. I was wondering why my mic wasn't working. I have done a little bit more research in terms of reading some papers, and I did find one that had like a pretty decent efficacy rate model. But other than that, I'm floundering a little bit, kind of gasping for air, so I'll probably need to come in for office hours.
I think we should do that sooner. I'm all of the projects are really good, but I'm particularly eager to get. I mean, I'd like to see I mean, I want to see all of these done in UPOS nanoHUB applications, but yours in particular, perhaps I think would be very fashionable if we got it done.
Mm-hmm.
I think this question about vaccine efficacy in age-structured populations is in the news a lot, and it's pretty easy. I think we could build a model of it in about an hour.
Right.
Now, finding the parameters for the model might take somewhat longer than that, but basically building the model that we've talked about, I think wouldn't take that long. So if we sat down for an hour, we could probably help you get started on that pretty fast.
Yeah.
Then there's a lot to do. I mean, the writing the model structure is not solving the problem, but at least it would give you something to play with. Let's make a point, but at that point. Okay.
Sure.
Say, how are you doing in terms of the model? We talked a little bit. How are you doing since then?
You said me, right? Sorry, you faded out a little bit.
Yeah.
I have I haven't done a whole lot since we met, but since the last time I shared with the class, I have now got a somewhat, or well, it's a working model looking at when radiation is being applied. I still need to go ahead and look back at the other paper to see where they got the equation that we were missing. But then definitely meeting later this week to make more progress on it. So.
And Jeffrey, were you able to find out what was wrong with going on with your system?
Yeah, I've gotten the parameters to look a little better. And I plotted all three of the plots together in the paper. They kind of eventually approach a limit cycle, but mine just kind of goes down, down and down. So there's something I still need to figure out with that, but overall it's pretty okay, I think.
Okay, so we should try to, again, get together and see whether we can try to debug that. Because it looked like you had oscillations from what we were doing before. But... Kali, I guess we were just trying to do sort of a quick check-in on progress on projects. And I was saying that Josh and I are available to meet with people later in the week, if that's helpful. So if you just give us a few second update on where you are with things, and what we could do to help out.
My few second update is that last week was kind of busy for me, so I don't have that really much to update. I have my 2 models that I've written, so right now it's just about heading underway with a little more intense with that parameter sweep and seeing how I'm gonna break up all the data that I have, how I'm going to like group states together into regions, and how I'm going to define like maximum number of vaccines, etc.
Okay, great. Well, that's fine. As I said, it's okay to say you didn't do anything. I just want to make sure I know where people are. In your case, it doesn't sound like, I wasn't saying you didn't do anything. I was just saying the check-in is really a check-in. So people that we've talked to, please try to make an appointment. We'll try to meet with people. And Josh, then I'll turn it over to you.
Sure, no problem. I want to reiterate on what James said in terms of reaching out if you need help with your projects. Don't hesitate to email me or email James, and we can schedule appointments. I'm pretty much free anytime after Tuesdays. Pretty much booked before that. So we're gonna come back to what we were our plan last week, which was to replicate results from 2 within host vital dynamic models. James, by the way, so how do you want to, we didn't actually decide if we wanted to split the groups or in the papers together.
Why don't we go through the material, discuss the models a little bit, what people think about it, and then we can let people in the class decide whether they'd like to split up into 2. I think 7 people in one group is too many.
Right.
So we probably need to divide into 2 groups. I think 3, but you could do 2, 2, and 3. So you could do 3 groups. Depends a little bit on how you feel about it. The question was, we've got 2 papers we were going to work on. Whether you think it's more useful to have everybody work simultaneously on one of them and make sure that everybody is able to replicate that one paper successfully, and then move on to the other paper, or
Right.
whether it's better to have one group work on one paper and the other group work on the other paper simultaneously.
I think it even if everyone works on one paper, still splitting the group into two is good, because then we can work. You can work with part of the class, and then I can work with the other one. Even though even if working on one paper at a time.
I mean, it might actually be better to have 3 groups.
Yeah.
Because 4 people is a little much, but it's up to you. You have to tell us.
But
how
I think we should split the groups, but have them all work in the same paper.
How do people feel about that? It's more a question of group size. We can either do one group of 4 and one group of 3, or 2 groups of 2 and one group of 3. I guess the people of the 3 are the same either way. It's more of a question of whether you prefer to be in a group of 4 people or two people.
Honestly, I prefer a group of 2, since there are 2 people who are knowledgeable in this chat. That would include you, Professor, and then Joshua. If there were 3, you would have to like simultaneously like shift between all the rooms.
Right. That's my concern as well. That then we might not have a resource for them.
Right.
Right. Although I think people could work. We'll see. Okay.
Yeah. Let's just walk through the papers, and then we can.
That's fine.
We have a little bit of time to think about it.
Again, it's really what works best for you.
Right.
We're flexible.
Okay. Cool. So let's just today we're gonna review what we were working on last time in terms of fitting the within host model to experimental data, and then after reviewing that, we will move on to try to replicate the papers that we suggested. Just as a reminder, this is the model that we've constructed. Again, the model was motivated by the main biological components or actors through the resolution of the viral infection. And then the choice of parameter values that we came up with were chosen such that the viral load curve resembled a viral load curve from experimental data, where it has different phases of the viral growth curve. But in terms of the components of the model has cells of or let's call them target cells of 4 different types, or in 4 different stages of infection. They can be uninfected, early infected, late infected, and dead. And then cells switch from uninfected to infected when they are exposed to the virus. Early infected do not release virus, but start releasing what we call the cytokine signal. So do late infected as well. But then late infected release virus to the extracellular environment. And then they can also die because of this viral infection. So there is one mechanism of death. And then we incorporated another mechanism of death that accounted for the adaptive immune response. We extended the model to incorporate the recruitment of these effector cells, which would be cells corresponding to the adaptive immune system. And the way that recruitment occurs is that the cytokine signal gets transported from the local tissue or the infected tissue to a lymph node organ. And then in the lymph node, does signal, and we discussed that what this actually represents is complicated, because the way that signal is relayed is both by the transport of signaling molecules, but also by some cells collecting information in the infected tissue and transporting that information to the lymph node organs. So this part here represents both of those signal transport processes. But the idea is that once a signal arrives to the lymph node, then that signal induces the proliferation or activation of the effector cells of the adaptive immune system. And then those effector cells are transported back to the tissue. And then when those cells come into contact with the infected cells, then they induce cell death as well. And we talked a little bit about some of the assumptions that we incorporated here. So for example, in this model, effector cells are able to recognize only the cells that are secreting virus, whereas in reality, effector cells are able to identify infected cells even when they are not releasing virus. So that could be a way of modifying this model, modify all the assumptions into this model. Depending on the level of generality, we could have cells interact with the virus if we wanted to as well, directly, depending on what we want to represent. But we wanted just to provide a basic representation of what it's called the cytotoxic effect of the effector cells of the adaptive immune response. So this is what it's represented here.
I was going to say another thing, one thing that probably really, another thing that's missing, which is probably pretty important in reality, is that when the infected cells die, that creates a pro-inflammatory signal. So there probably should be an arrow from this I2 goes to D that points at C.
Right. But
and the effector cells themselves also secrete a pro-inflammatory signal. So there should probably be an arrow from E to C.
Right.
That would lead to stronger feedback.
Right.
Positive feedback.
Mm-hmm. Yeah, that is a good point. Especially not so much concerned with the death signals because I mean that's but the fact that this has a independent dynamics would imply that the amplification of the signal would just not adding, but it might have an actual different dynamics. Would just not be additive, but it actually behave differently, which would be important. What about, and there's a lot of other components that were not necessarily included here, as I mentioned, in terms of the adaptive immune response, all we incorporated into this model was the cytotoxic effector cells, but we could have included the antibody response as well if we wanted to, right? The effector cells of the lymph node, they can be of two types, right? They can be the cytotoxic type, but they can also be the antibody secreting type. And then we would have a different type of cell in the lymph node that then secretes these antibodies. And then these antibodies get transported back to the tissue the same way that the cytokine here or the signal is transported to the lymph node. And then they can bind to the virus and disable it. And they can also reinforce this cytotoxic activity of the effector cells. So yeah, all of this is to point out what are the assumptions that we incorporated into the model, what are the basic ideas that we left out, and why are the reasons that we chose the components that we chose. So when we simulate the model, we talked a little bit as we were walking through the model. I'm not gonna do it again, but we talked about the delays between these different curves and what the curves are telling you. But the basic idea, the basic idea is that we chose the parameters, the values of the parameters in our model, such that that the vital load curve reproduced this, or at least had this, these 4 stages of vital growth. A state where an eclipse phase where no virus is present in the extracellular environment, although you have although some cells are infected. Then a phase of rapid exponential growth. Then a saturation part of the virus. Then a slow clearance that we associated with the innate immune response, and then a rapid clearance that is associated with the adaptive immune response. And that's the parameters that we chose. We chose the specific values. We chose them so that we got an approximate, not just the 4 phases, but a quantitative approximation to those. So we wanted this exponential phase to occur over a period of 2 days and increase your vital load from 10 to the 1 to 10 to the 6. And so we wanted the slow clearance to occur over a period of 7 days and go down from 10 to the 6 to 10 to the 5. And then we wanted the rapid clearance to occur over a period of a day and a half and go from 10 to the 5 to, let's say, 0. So we chose the parameter values in a way that would approximate those values in our curve. And then now, this was this is basically what I explained. But the values that were motivated by an actual experimental vital load curve, and we used this to estimate what were the different rates at which all of these phases were occurring. Now that then we talked a little bit about how these vital load curves are measured. In general, the idea is that you have a sample where you have a sample that contains your virus, and you want to know how much virus is in that sample. The way you do it is that you basically have a plate with cells that are susceptible to be infected by that virus, and then you create a serial dilution of your sample, which means you basically add water in sequential steps, and then you infect your plates, and then you count how many of your cells get infected, either by looking at the plaques, which is what the plaque assays are, or by other means, immunostaining, for example. You can count how many of your cells were infected if they don't form plaques, because it depends on your specific experimental setting. But the idea is that then you can quantify how much virus you have in a sample. And one of the exercise in the homework is to explain one of the papers that we looked at, that I suggested, to explain how did they actually measure this vital curve. In in this particular example, we looked at the paper and we talked about how the mice were inoculated, and then mice were sacrificed at this specific time point, and then their lungs were digested, and then the supernatant was used to do the plaque assay. So that's basically how they measured it. It sounds easier than it actually is when I describe it like in one sentence, but in reality, it's a very extremely complicated, time-consuming procedure to measure vital loads. So once we had our model, so from here we wanted to compare how well our model was, how closely our model was to the experimental data, and then we wanted to fit the model such that we get a better approximation between or a better values of the parameters that reproduce the actual experimental curve. We spend the rest of that class just looking at the fitting algorithm that we've been using in the class. We loaded the data. We specified the model as an anti-money string with the parameter values as we estimated them. And then when we compared the data to the model, we saw that even though we are close, it doesn't exactly reproduce the experimental data. So if you wanted to more accurately reproduce that experimental data, then we have to fit the model to the data. And then, we talked a lot. We reviewed what the fitting is trying to do, right? So basically, the idea is that when we created the model and we estimated the parameters, we did it such that our starting point is near. So the idea of fitting is that you want to minimize the difference between your model prediction and your experimental data. And these values here represent that areas in the parameter space, or for combination of parameter values that minimize that difference. When we chose the parameter values that we chose, when we were putting the model together, we came up with values that bring us closer to that minima, but it's not exactly at the minima. That's why we see that difference between the model and the experimental data. But the reason it's important to start with that good approximation is because, especially with this model, the rest of the parameter space can be really flat. So imagine this being really flat in all directions. And then if you start at a random point in that space, the probability that you're gonna end up in a area that you're gonna actually find the minimum is actually quite small. And there are algorithms that are designed such that you can start pretty much more or less randomly, and they will try to find the minima. But with some of the stuff that we've been working on with James, I've been working up with James, is to come up with algorithms that are more robust. So you don't necessarily have to start, but especially the models can get very complicated, and then the algorithm is exploring the space can take a very, very long time to find, if they find it at all. And this is what James has been saying, that when James points out that fitting is as much a science as it's an art, it's because it requires a little bit of intuition to be able to start at certain points to delimit what is the space that you want your algorithm to explore, such that you can actually find a parameter space, a minima of, or a good approximation between your model and the experimental data. So, and then we walk through what the algorithm is doing. I'm not gonna work through it. I'm not gonna walk through it again, but the idea is that we're basically measuring in line 12, we're measuring the distance between our model and the experimental data. And then that is the way we're quantifying how far away we are from how far the model is from the data. And then this is a function that the optimizer is going to try to minimize. And we talked about if we needed to incorporate a second data set, we would add it here. And we discussed a little bit of well, what it means to add a second data set, how you weigh the importance of one data set versus the other. How do you have to normalize it to account for bigger difference in one data set versus the other? We talked a little bit about that. Maybe we can come back to that later if we take a whole class in fitting. That's something we can maybe come back to. And then we show the basics of using this wrapper. It's called LimFit. That is a package that allows you to easily define what are the parameters that you want to fit in your model, what are their starting values, and their minimum and maximum values that the optimizer is going to explore. And then we run the optimizer, and we let it run for a certain amount of time until we got a better. We started here, and as the optimizer ran this way, we got a better approximation of our model to the data. And then we talked a little bit about when to ask the model to stop and so on. And then let me see. Okay, yeah. So then we looked at how the results compare before and after our models were fit, and then we spent some time playing with the model, constraining the optimizer such that we could get a model that fit the data, but also resulted in not all of your cells getting infected and dying at the end of the simulation. So that's where we ended the class last time, right? We were trying to come up with a good fit to the experimental data, but we added an additional constraint. Instead of just getting a fit to the data, we also wanted to have some cells survive at the end of the simulation. And that's, we just spent a little bit of time playing with at least adjusting the algorithm, the code, so as to add a different constraint. We saw that it was not straightforward. It's not like the model can in fact produce you can find a parameter set values that will result in your model, not of all of your cells dying or getting infected by the end of the simulation, but then it's not necessarily a good fit to the experimental data, to the observed experimental data. That's where we ended last time. So before I move on to the replication part, any questions so far? Because in the homework, I'm asking you to take a different experimental data, and then fit the model to that experimental data, and then compare the parameter values between one fit and the other. But I wanted to make sure that the question was that the problem was clear in terms of what I was asking. And if you haven't had a time to look at it, it's fine. But the idea is that instead of we fit the model to this experimental data. So what I'm asking you for the homework is, I'm giving you 3 different data sets. I'm asking you to take this model and fit it to those 3 experimental data sets. And those 3 experimental data sets correspond to 3 different groups. What they did is that one group is a control group where they were infected by the virus, and that they let the infection run its course. Then a different group corresponds to people who were treated early with antivirals. And then a third group was a group that was treated later with antivirals. And each of those 3 give you a different curve. And the question in the homework is, and I'm actually not I haven't looked at it myself, but the idea is whether we can, by looking at the values as they are fitted to the data, whether we can relate treatment to some modifications of the parameter values of the model, because that's basically what the way treatment is often modeled in this type of within host models is by modifying certain parameter values, by adding certain modifiers. They usually are added explicitly, right? So they have, okay, I'm going to multiply the viral production rate by a factor epsilon, and then the value of that epsilon represents the effectiveness of my treatment. But when you get the fit, you should be able to see a difference between these 2 groups in terms of P, and then that difference tells you what that epsilon value is, or what the effectiveness of the treatment is. So by looking at how the parameter values change, you can get an idea of how treatment is altering or affecting the parameter values in your model. That's a point of that exercise in the homework. Okay, any questions so far? Okay, then we can start. The idea today is that we're gonna try to replicate 1 or 2 within host viral dynamic models. Right, originally we wanted to split the group in 2 and then have you discussing in the group what the model is about, what is the problem that it's trying to solve. Then implement that model as an anti-money string, simulate it, and displays the time courses such as they do in the papers. And then before doing anything else, just change a couple of the parameters to get an idea of what the parameters are doing. What happens if my infectivity increases? I should see my cells getting infected more faster or more slower. If the virus is produced more faster. So just to that is a good strategy to understand this without doing a systematic analysis, at least get an idea of how the model behaves. What are the parameters? What is the effect of changing certain parameters of the model? And then after that, we will do this bifurcation analysis, which is done in both of the papers, and that is something we haven't discussed, right? But the basic idea is that you take your model with the parameter values, you run it, and you see what happens after you run it for a long amount of time. We call it what happens at the end. And then once you've done that for a set of parameter values, then you change your like a critical parameter, let's say, your infectivity, and then you see what happens at the end. And what we saw in the SIR models, right, is that when you increase R naught or you decrease R naught, you go from a higher proportion of your population getting infected at the end. And then as you decrease R naught, that proportion decreases, and below a certain point, your population doesn't get certain amount, almost no portion of your population gets infected, or the infection doesn't get hold on your population, right? So that is the idea of the bifurcation analysis, where you take one parameter value, you change it, you change that value, and then you see what happens at the end, and you compare the end results for all the values of the parameter. James, do you want to say something else about that bifurcation analysis? I know we haven't discussed it like in detail. I'm just trying to explain it.
Well, I mean, a lot of the math in the papers, which I hope people skipped if they found it unpleasant, was trying to find those bifurcation results analytically. And there's some basic rules about when, for example, the steady state goes from being stable to unstable, which we haven't gone into in detail in this class because of the mathematics involved. But nevertheless, we've already seen this to some extent in our SIR models. If R naught was less than 1, the disease died out.
Right.
If R naught was bigger than 1, the disease propagated. And then in some of the models, everybody died from the disease. In other models, we were left with a situation where some people survived and some people died. So you might have a bifurcation. So you always have a steady state, which is if there's no virus and nobody's sick, then you wind up with everybody well, everybody stays susceptible. So there's always a steady state with nobody sick. If R naught is less than 1, that steady state is stable in the sense that if I introduce a few sick people, the disease spreads a little bit, then goes away, and I go back to everybody being okay. If R naught is intermediate, then I could have a situation where I have what's called an endemic, where I always have some people sick, but not everybody. And in that case, the state with nobody sick is unstable, because if I introduce one sick person, the disease will spread until there are a reasonable number of sick people. If I start with a lot of sick people, the number of sick people will go down until I reach that equilibrium number. And so then that's a steady state. And if I make R naught very big, depending on the model I'm using, I could have a situation in which everybody gets sick or everybody dies. And clearly, once everybody's dead, I'm done. And so I could wind up with a situation where the stable steady state was everybody dead, for example. And so there, depending on the model, you could have multiple transitions, but the one we saw the most was between R naught less than 1, where everybody healthy is stable steady state, to R naught greater than 1, where some people sick is the stable steady state. And we'll see that in a few minutes, because the example we're going to do will have we'll have 2 transitions. And so that'll be an example of that.
Yeah. So and we'll work on this together, and it will become more clear as we work on it.
I was thinking logistically, you've got this nice layout here. Maybe we should actually have this sort of formalized, where we say, okay, everybody goes off and spends 15 minutes working on the number 1, and the group writes up a paragraph for 1 slide or 2 slides, right. And then everybody comes back and the 2 groups present, and then we do part 2, everybody goes off for some time and come back And does that seem like a sensible strategy?
Yeah, but do you still want to do the two different papers, or do you want to do the same paper both groups?
Well, why don't you walk through the two papers quickly, and then we could have people vote.
Yep. And
I'm
then
okay with it either way.
Right. Okay. So the first paper is I'm sorry, the title is wrong here. This is the hepatitis V infection model. So in this model, they are trying to they model the infection of in the liver by the hepatitis B virus. The idea is that they have some function where cells recover after they die, which is an appropriate representation of the liver. And then they go through infection and they release virus. But the idea in this paper is that as I change the parameter values, specifically R0, I go from this is the old one. So I go from a regime where I get no oscillations, or the oscillations die out, to a regime where I have stable oscillations, where my cells get infected, die, but then new cells come in, get infected and die, and then new cells come in, get infected and die. And then we can explore how the transition between this state where I have no oscillations to a transition where I have oscillations by changing the critical parameter R0. This is the new, that was the new, I had an old version. We will come back to this, but this is what it's called a Huff Bifurcation, where before a specific value of the parameter, you have this decaying oscillations or damped oscillations that the system starts oscillating, but eventually settles into a fixed point. Whereas in the after you increase the parameter value, then you what you get is stable oscillations, and then we can measure how fast those oscillations are decaying, and we can measure how the period of those oscillations is changing. We will do that as part of that exercise.
It occurs to me there are two things we could add to this slide. One of them is the definition of R0,
right,
which is beta times gamma divided by A times mu.
Right.
And then the other thing you could actually measure, which would be interesting, would be once you have the bifurcation, what the amplitude of the oscillations is.
Right.
So we said measure the decay of the oscillations for R0 less than 7,
and then
measuring the amplitude of the oscillations for R0 greater than 7.
Right. We can do that. That's definitely work covered.
And then the period and the other thing that in that part 3 and this figure and this graph this diagram is meant to illustrate this here, is if you start with oscillations bigger than the equilibrium ones, the amplitude of the oscillations goes down, which is what you're seeing here. But if I started here in the middle, this is what problem 3 is. If I start here in the middle, the amplitude of the oscillations goes up. And so that's something you should be able to see actually in your simulations.
Right, right. Who was talking about the limit cycles? Someone mentioned that they had limit cycles on their model.
That was me.
So yeah, this is the same idea here.
Yeah, this is classic. It's a little simpler than the ones you get in neurons because you don't have the spiking that you get in action potential. Same idea. The other thing is you actually have three bifurcations here. If R naught is less than 1, then all of the infected cells go away and you're left with all X. Y goes to 0. So X, you should probably have written that too. X is the uninfected cells. Y is the infected cells. If you go to above 7, you get these oscillations where the disease comes back, cells die, comes back, cells die. And then if you go above 9 here, sort of surprisingly, the oscillations go away again. And almost everybody's sick, which is what this blue curve being small means. And if you go above 10.5, all your cells die. So you actually have a transition at 1, a transition at 7, a transition at 9 point something, and then a transition at 10 point something. So in this first region, so this region here, there's no, this region is Y equals 0, no dead cells.
Right.
This region here is Y equals Y star, X equals X star, fixed values.
Right.
This region here is oscillating.
Right.
Between X and Y. This region here is stable again, but smaller values of the uninfected. And then above here, that you have everybody dies.
Right.
And so you actually have, this is a nice example because the 3 equations look fairly simple.
Mm-hmm.
The first term of this recovery model is called logistic growth. I've had slides on logistic growth because that's usually the very first model I do in class. And they kept getting delayed because we weren't doing that kind of population modeling. But everything else in this model is pretty simple.
Right.
And so the fact that it has so many interesting behaviors is rather nice. The paper itself is a rather heavy read because the analytic solution of these is not very pretty. But the numerical solution is quite easy. And so, okay, turn it back to you, though.
Yeah, no, yeah, as you said, it looks simple in terms of the equations, but in terms of the behavior of the system, as you change the parameters, you can see that you can get a rather complicated behaviors. But again, I did wanted to point out that when we are talking about the bifurcation diagram here, right? What we're looking at is what happens when you run the equations for long enough, when you run the system for long enough, right? So here we know that this oscillations are damp, but we looked at what's happening at the end, right? And yeah, and actually that's on the paper, James. The definition of R naught. I'm sorry. Oh, I cannot modify now. Right. And then the next paper, which is actually the HIV competition model. That one is the same idea, right? They are looking at what's happening when you they implement the model. They show what happens with the model when you just run it with the base set of parameter values. And then what they do here is basically changing 2 parameters at the same time. In the previous one, we were changing 1 parameter, which was R naught, right? And we were seeing how the system behaved. In this case, they are changing 2 parameters at the same time. The R naught for one virus versus the R naught of the second virus because they have two viral strains. And then what they're showing here is what happens, basically what virus is dominating over the other one as you change, simultaneously change these two parameters. But what I would suggest, if we choose to replicate this paper as well, what we were talking or suggesting is that instead of doing this image which will require you to modify two parameter values at the same time, we can fix one, right? Let's say RS here too, and then change RF and see what happens in a line here. And we should be able to see what happens in these three regions. And then choose an RF as fixed RF and then change the RS through a line here. And then you should be able what each of these regions here are representing by looking at the specific behaviors of the system in those three regions. So you should be able to replicate 3 plots in this area and then 3 plots in this area that show basically the different behaviors in the regions, in the 3 different regions.
Again, I'm sort of doing that first part, which is explain what this means, but I'll talk about it a little bit, then you can think about it more, which is, suppose that you have 2 mutations of your virus. Here they're called S and R. It's not likely that, all things being equal, otherwise that those 2 viruses are going to replicate exactly the same rate. One of them will replicate a little faster than the other because they're different. And so RR and RS represent the R naught for each virus separately. So if I have a big value of R sub R and a little value of R sub S, that means R sub R replicates faster than R sub S. If I have a big value of R sub S and a smaller value of R sub R, that means R sub S replicates faster than R sub R. Well, you'd expect that whichever one has the faster replication would win at some level. That may or may not be true. That's the first thing you could check. The second thing is, so in particular, if the R naught for either virus is less than 1, you expect that virus to disappear. So down here in this region where RR is less than 1, you expect the R virus to go away. If RS is less than 1, you expect the S virus to go away.
Right.
In general, you'd expect that whoever has the bigger value would win. So if you're under this curve, under this diagonal line, RR is bigger than RS, so you'd expect the R virus to win. If you're above it, RS is bigger than RR, so you'd expect RS to win.
Right.
In fact, what this is suggesting is there are regions where they both coexist. And that's because it's not quite as simple as them being equivalent and competing directly.
Right.
But so that's the first thing you could do. The second thing is you'd say, well, why do you even care about whichever one reproduces more slowly if it doesn't win? And the answer there is that suppose that the one that reproduces faster is more susceptible to the drug you're using to treat the disease.
Right.
So now I come in with the disease, with the antiviral. I apply the antiviral. I kill all of the RR, for example, but the RS stick around and come back. And so that would be an example where the use of the drug is selecting for the strain of the virus that normally would be selected against.
Right.
And if the drug is very effective against one strain and not at all effective against the other, then the problem is that as you keep using the drug, you select for the resistant strain and the drug stops working.
Mm-hmm.
And this is a classic problem with chronic diseases. acute diseases where it goes away, it's not so much of an issue. But chronic disease like HIV, where you live with it forever, so hepatitis, this is a problem. With cancer, it's a problem. And so this is a nice example of this kind of strain selection. In this case, there are only 2 strains, R and S. But in principle, you could play the same game again and again with different strains.
And they do have in this paper a part where they incorporate treatment and then they see what happens at the end, which both viruses, which would be also interesting to try out. Okay, so yeah, so those are the 2 papers that I suggest we tried. So how do we do it? Depends on how, yeah, it's up to us now. We have 2 hours. So first, let's create at least 2 breakout rooms. But that's, James, you have to do that.
The breakout rooms exist. I haven't put anybody in them.
Okay, so
I would suggest we just go down the list. I'd say Antonio, Devin, Tyler, go in breakout room 1, and Brian, Mercedes, Jeffrey, and Collie go in breakout room 2.
yeah, that's fine.
And maybe the first thing to do is to have you guys break out and do that. Why don't you spend 5 minutes looking at the papers, both papers, and come back and tell us in 5 minutes whether you want to do, which paper you want to do. Is that all right? The problem with the breakout rooms is going to be the usual one, which is that the person who's hosting the meeting, which is me, we lost. Who did we lose? We lost. Devin already joined. So we lost the person who. So did that work? You could join. You should be able to go in and out of the breakout rooms on your own. I set it up that way. But the problem is that as the host of the meeting, if I join the breakout room, bad things happen to the meeting. And so I guess I can dive in, but it may be easier that if you guys go into the breakout room, if you need help, send somebody out and ask me, and I'll come back. Okay.
Or you can say, or one group can stay here, and then you send the other one to the other breakout room, and I go there. And so we just use one breakout room.
Or for that matter, to begin with, I think it's better for the groups really should try to do so as much as they can on their own.
Yeah,
so why don't we start out with you guys splitting up, spend 5 minutes looking at the 2 papers,
right?
Come back, and then we'll go through this list of things to do. And Josh can dive in and help you individually in the breakout rooms. And if you need my help, you can come out. So somebody has. Unfortunately, because of the way Zoom works, it's a problem if I join I suppose I could switch off. Either Josh or I could be in the breakout room, but not both of us at once.
Mm-hmm. That's fine. Yeah.
Just because of the organization.
Yeah. That's
fine. So shall I assign? How did we say? Said Devin was going to be in room
so
1.
Devin, Antonio, and Tyler in 1, and then Mercedes, Callie, Brian, and the other one.
I don't know how to join a breakout room on.
Okay. But you still have to assign me, because I don't.
The problem is going to be to get you back and forth between the 2. I'm going to need to move. I'm going to have to move you, I think.
Right.
I made you co-host, so you should be able to join.
Yeah, I'm co-host, but let's see. Yeah, I'm co-host, but I cannot go into the breakout rooms unless once you assign me to one, I can move between them, but you still have to assign me to one.
So do you want me to put you into one of them now?
Yeah.
What I was going to say is, why don't you go and tell people? I'd sort of prefer that at least one person do that first oscillating model.
Mhm.
So I'd rather that they don't both pick the HIV model. But
Right.
if they want to one. Hey Antonio, what's up?
Hey, so just to clarify, these are two papers that are in the lecture 12 demos on the Google Drive, correct?
Yes.
Okay, and so they're also the same, like the two papers are also included in the homework?
Yes.
Right.
Okay.
So you may have already done almost everything that needs to be done, in which case, I guess the goal is to help those of your classmates who didn't get so far, or to
Okay.
go further with it.
Yeah,
Which-
you already, some of the stuff that you already showed, that covers 1, 2, and 3.
Right.
So
Okay.
that was the idea with the homework.
Okay, I think
homework
we're
was
gonna go ahead and
basically-
do the first paper then, the HIV modeling.
Okay.
The HIV modeling.
Mhm.
I guess we didn't name the paper. So the HIV modeling was the one on the viral competition.
Right.
Yeah. The other one was okay. So you want to do that? Okay. So that's fine. In that case, we should probably push room 2 to do
The hepatitis.
push room 2 to do the hepatitis one.
Okay.
So I'll put
I'll
you
go-
into room 2 and say that they're sort of by default are being pushed that way. Okay.
Right.
Now, can you go see if Antonio, see if you could get back into your room?
Yep. Yeah, I can.
Okay, great. Okay. So in that case, what? Okay.
Okay, I told them to look at that paper, and they're gonna come back to when they're gonna focus a little bit on reading it and come back and talk about what the paper is on.
How long should we give them to do that?
You said until 6:10, so 3 more
Oh,
minutes.
but that was to pick the paper.
Oh, so yeah, then ask them to come back and then.
Or I could just tell them to keep working and do the
Right, to work on part 1.
right. But how long should that take?
10-15 minutes, and it shouldn't take too long.
625.
That works. Yeah.
Boy, these messages can only be something like 50 characters. So. You can edit, I suppose you can edit your slides that fix the typos you've got.
Yeah.
Probably. Excuse me, I'm back in just a second. Okay.
Yeah, that's fine. Okay. James, everyone is back, so we can.
Okay, so let's hear from group one. How'd it go? I guess Josh, you'll have to unshare so they can.
So, well, we don't have group one, number as by the number of the paper. So group one is the HIV model, and then group two is the hepatitis B model.
Okay, we can go first then. So reading through the article. I guess I forgot to type out the problem statement, but generally it was implementing an HIV infection that also included latent infection as well as a mutation rate. And so we implemented the latent and mutation applied HIV model and printed out the time series, which agreed with the researchers' time series. And then we started working on applying the reverse transcriptase inhibitor interventions. And we were still kind of coding through that. But the researchers' goal of the paper was to basically see how effective this intervention is in eradicating an infection of HIV. And they concluded that it wasn't effective enough, as the virus is too prone to mutate. But we'll see. We'll play around with the parameters and see if we can get it to be able to eradicate all of it.
I guess my main question is in terms of how do they represent the intervention? How is the model modified to include that intervention?
So they include
reverse transcriptase inhibitor.
Yeah, so they include this epsilon rate that basically reduces the infection rate. And then the S is for the wild type. And then they have a different epsilon R for the mutated. And the researchers claim that the mutated virus shows about a resistance to the inhibitors of about 0.5 magnitude. So it's half as effective. The inhibitors are half as effective on the resistant virus. So EPR is really just equal to half of epsilon S.
Okay.
And how do you apply the treatment? Do you have a event that turns the treatment on and off, or how do you?
The researchers just incorporated it into the model.
So it's always there.
Yeah, I didn't see any event where they introduced the inhibitors. That could be something that would be easily implemented, though. Something we could change.
Yeah, it's interesting because the way they implement it, well, because the treatment is reducing the transmission rate, right? Which is what E3 is doing here. In both cases, E3 and 10. Oh, well, E3 and E6. But it's also because you have mutation, right. So it's all of it is in the mutation rate. Whereas I would think of a reverse transcriptase inhibitor. Yeah, I mean, I guess that's one way of putting it, right? Because the reverse transcriptase inhibitor will prevent the cells from becoming infected even after the virus enters the cell. Whereas I tend to think about that step, representing that virus being able to enter the cell in the first place. But yeah, we can discuss that later. It's not important. It's just a way of how do you think in terms of how to represent your intervention in the model.
Right.
So it looks like basically that it's reducing the effective amount of virus.
Right.
Right, you're multiplying V by 1 minus CPS everywhere.
Right. Whereas I would probably represent it as preventing the transition from latent to late infected, and then having a reverse reaction going from latent to uninfected.
Well, I guess the question is, did the paper do it this way, or did you come up with this?
The paper did it this way. They
Paper did it this way. Okay.
yeah, they claimed that it reduced the virion production, and also reduced the infectivity of it.
Right, but you don't. It's actually not. If you look at lines 20, E15, and E17.
Yeah.
It doesn't seem to be reducing the rate of virion production.
Right.
Yeah, we noticed that too when we were looking at the altered differential equations that they presented, that they didn't actually alter the production of the virions.
I don't know. I mean, if you're reproducing their paper, you have to do what they did. But one thing to think about is in that first part where we're talking about thinking about what they did is trying to understand why they did that.
Right. I guess if they're essentially reducing this transition to infected, like wild type infected and wild or mutated infected,
Right.
Since those terms are included in the production,
Right.
kind of in a backhanded way that they're altering the production, but not directly.
Right.
I mean, again, the antivirals work in all sorts of different ways. And so it's certainly possible this one works by basically making the cells less likely to get infected when they're exposed to a given amount of virus.
Right.
I mean, PrEP works that way. I mean, essentially, that's how PrEP works for HIV. Right? People take the medication before they're exposed, and when they're exposed, the probability of infection is lower. The one thing that surprises me on the treatment side, I guess, if you're modeling PrEP, then you assume that the individual is on the drug before they're exposed, so it's prophylactic use. But and then I suppose it's a STD. If there's sexual contact with somebody who has both the wild type and the mutant, and somebody is exposed who's on PrEP, then the PrEP would be less effective potentially. So that this would be a model of that, I guess. It's a little different from a situation where you're already infected and then you go on the drug, which would be a model of how resistance develops within somebody who's being treated. And so, because initially, if I look at your graphs of time series, if we scroll up and look at the time series that you did, right, initially, the viral loads are all very low. And so if the treatment were very effective, the viral load should just stay low all the time. And so that's why I was sort of surprised. I would have expected, for example, that you wait until the viral load goes up, you're going to say 10 to 100 days later, and now you treat and see what happens. So you might want to see what happens. We did this a little bit, although we got the time wrong in one of the homework assignments just a week ago, where we said, okay, that was in the dengue model, where you say, okay, treat at different times. The only problem was we said treat at day 10 when the infection was already over. We should have said treat at day 4, day 5, day 6.
Right.
But you can certainly apply an event
of,
where you turn the treatment on.
Yeah, we can compare introduction of the treatment before infection, and then probably, I don't know, from maybe day 8 or so. Introduce the treatment there.
Late.
Yeah, see what happens.
And one a day 100 and see what happens.
Mm-hmm.
I mean, HIV, one of the issues, of course, is, historically at least, is that people weren't diagnosed. So they'd live with it for years before they were diagnosed. So you could be in a late stage of infection before treatment.
Right.
So now those are some interesting things to do. So you're already quite far along here. I would encourage you to put some text into your notebooks. You could, I don't know if there's some way you could share the notebook so you could work together on it. so everybody can contribute. But I mean, this would be a nice, again, I'm always thinking of making this into a little nanoHUB application. So you would like to have some text that explains what you're doing, what the model is. And then in your example, you'd have some parameters you could change. We haven't shown you how to use sliders to change the parameters, but once you say which parameters to change, Josh and I could show you how to make that, put that into a slider so that if you made it a little application, somebody could say, well, now I'm going to change the dose of the drug and see what happens. Now I'm going to change the time at which I give the drug, see what happens. I'm going to change the relative efficacy of the drug between the two viral strains, or the R naught between the two viral strains.
Right.
Okay, so that's great. Great. Okay. Anything else you wanted to discuss so far? I mean, you're very far along already, so that's. And I hope everybody in the group is able to do this together, because it's important that
Yeah,
some
and,
people.
and we still we'll still need to work on the on those bifurcation diagrams anyway.
Yeah.
So we can work together.
Okay. Okay. How about the other group? How's it going? Somebody going to be spokesman for the other group?
Let me see.
Or spokeswoman? I guess spokesperson.
Right, so we didn't get as far as the other group, but we have some stuff down. So this is the hepatitis paper. Hold on. Anyway, so after looking over the paper, we found that the purpose of the paper is to model the infection of liver hepatocytes by the HBV, but it wants to create a more experimentally accurate model of this infection. So apparently other models, they assume mass action, and there was a second thing where they didn't have, where they had the healthy hepatocytes growing back at a constant rate instead of, but apparently these are not what is seen in real life. So they changed their model by swapping out the constant growth rate for the hepatocytes with the logistic growth and using an incidence function instead of mass action for their model. So we haven't gotten to the coding portion yet, but we have, and we have the equations on one side here. That's the model taken from the paper, the one we had to do for homework. And then to the left, we have something of an arrow diagram, or I suppose it is an arrow diagram based on what we have in the equations. And then obviously we haven't simulated the model yet, but we do have the expected results ready, so we can make a comparison.
Okay, I can see one small thing, which is, since you're creating X, the X should be on the right-hand side of the arrow instead of the left.
Right. That's important.
Just as a small. So and I should maybe say something, since we haven't done logistic growth. A couple of things. The liver, of all the tissues in your body, the liver is one of the very few that regenerates. If I cut your arm, if salamanders and some fish and some frogs are very good at regeneration. If I cut the arm off of a salamander, it grows back. However, and some, of course, when you're wounded, most tissues will heal. But if I cut an arm off of a human being, it doesn't grow back. Winds up that humans have a little more regenerative capacity than you'd think, that if you cut off the tip of a finger at a child before the age of about 18, it will grow back. So that's salamander-like, but it's still pretty limited. Most of our tissues and organs don't have the ability to rebuild themselves if they're mostly destroyed. If the tissue integrity is destroyed, you can't really rebuild it. Liver is a little bit different because it doesn't actually, quote, regenerate. It's what it's called hyperplasia. But in any case, if I cut out 80% of your liver surgically, 4 or 5 weeks later, your liver will be back to normal. And so the liver of all the organs in your body is the one that recovers the most rapidly after injury. It makes it a little bit mysterious why liver failure happens, because sometimes the liver gives up and says, I quit. But in terms of surgical resection or cell killing, the liver is able to compensate rapidly and completely. And so this idea that the liver after injury grows and recovers to its original state, which is what the logistic equation does, is reasonable. So that makes sense to me. If we look back at the equations, so that if you look at that first dx by dt equation, the first equation is simply saying that the liver stops growing when it has a volume of k. So the growth rate of the liver is Rx, so it grows exponentially if it's small, and then the growth rate slows down until t equals k, at which point it stops growing. So that's a classic growth and recovery model. And then you said that the infection rate depends, is a function of is not mass action, and you have bx over t. And what that's saying is something actually which is interpretable, something to think about, which is if x is the number of infected cells, actually, is it bx? Shouldn't there be a v in there somewhere? Yeah, it's got in your x goes to y, you have the v. It's missing from the equation on the right. And I don't want to spend too much time on it, but if I want to make sure that you don't have a typo in your equation, so that when you turn it into antimony, there's a problem. So if you look at your equation, your arrow diagram, the rate at which x goes to y is b times v times x over x plus t plus y. So v over x plus y is just the density of virus in the liver. And so this is mass action. BVX divided by t is mass action, where you're using the density of virus as your rate of infection. So that is an arrow diagram that makes sense. Then you have linear death of the infected cells. That's minus AY. You have the infected cells, the Ys produce virus at a constant rate. That's linear. And they die, and the virus is cleared at a constant rate. So the one thing you might want to put into that slide also is that R naught, which is the R naught is increases when the amount of virus being produced increases. So R naught would be bigger when gamma is bigger. It's bigger when the infection rate, B is bigger. So R naught goes like B and times gamma. And the faster the virus is cleared, the smaller R naught. So it's divided by mu. And the faster the infected cells die, the smaller the R naught is. So it's divided by A. So R naught would be B times gamma divided by A times mu. And that might be something you'd want to put on that slide. So, but that's fine. I think you've got a start there. So Josh, how do you want to go forward? Do you want to break up again and start trying to get this coded up and replicate those time series?
Yeah, so I would suggest that you work with the people with the hepatitis B group, and then you send me with the group working on the HIV, and then
Okay.
you maybe they can stay here and work with you, and I'll go to the other breakout room.
Okay. Okay, that's fine. I'll stay here and we can talk together about the hepatitis one. Okay.
Yep.
And shall we say, well, how long? We've got an hour and 10 minutes left. So
Let's meet up again in half hour and see where we are at.
okay. That sounds good. So can you join, or do I need to move people?
No, we can join now. I'll join. Yeah.
Okay. Can you go to room 1? All right.
Yeah.
Okay. So why don't the room 1 people, Antonio, Devin, and Tyler, go back, and Josh, and let's talk together with Kali. And I don't, and maybe you want to screen share. I don't know how you've been dividing up effort between the 4 of you. So you have to tell me how you've been working on it. I can just sit back and listen while you talk. But if you want my help, I'm available.
Sure. I guess we'll just ask questions as they come up.
Yeah. We just kind of discuss among each other usually.
All right. So the first thing we have to do is change.
Just code this up, or change the antimony diagram, right?
I think it's important that you probably you correct. I don't know whether you had that, whether those typos were just in the slides, or whether were you working with a Tellurium notebook, or how are you doing it?
Oh, the typo there, that's actually from the homework.
Okay, so there may very well have been a typo when Josh typed it up in the homework. So I encourage you to go back to the original paper.
Mm-hmm.
So
paper has the V in it.
So I would suggest that whoever is sort of the scribe, whether you're using the Tellurium notebook or I suggest everybody have a Tellurium notebook open, and also the paper open. And I'd suggest also probably doing the split screen the way I did it in class, so that you can have the paper on one side of the screen and the Tellurium notebook on the other. And then whoever is sort of the reporter can type it as you discuss it, but you can also each do it on your own as well. That's, I find, the most effective way to do it. Is that you don't have to do it that way, but that would be my suggestion. Then I'd suggest sort of the reporter do a screen share so everybody can look together.
Okay. So do you guys just want to work on the model on our own first and then come together?
Let's attempt that first.
Okay. Does everybody have the paper?
I do. I think everybody else does
so.
as well.
Okay. So I'd say then the thing to do is to try to basically, I mean, the strategy you showed in the slides was perfect. It was more a question of just making sure that all the terms in the equations are the ones that you want. And so I'll again, I'm happy to help out, but I'll sit here quietly, and if you ask questions, I'll be available.
Okay, I have my equations written if anyone else is ready. I haven't tried to simulate or anything. But it's written.
I'm not quite done typing, but Callie, if you want to go ahead and just kind of be the person who's the scribe, that's fine.
Sure.
I appreciate people's patience. It's a little awkward doing this by Zoom. It's easier if you have everybody in tables in a room, but we'll figure it out, I guess. So I guess, Kali, if you want to screen share at some point and show us what you've got, that would be great. No rush, you decide when.
Okay, I think I'll wait to screen share until after I have everything written and not just the equations. Does anyone know what the initial condition for the amount of free virus is in this model?
That's a very good question. My experience is that it doesn't matter much. So you could set it to any small number and it should give you the same result.
Okay.
At least when I did, when I saw, I did actually do this, Josh and I did this problem together before we assigned it. And we checked that the initial amount of virus doesn't make a big difference. You can make it just a number like 1, something small, or even 0.1. Looks like my cat may come to check out this class.
Well, at the moment, I've only tried to replicate the first figure. I haven't gotten around to plotting the percentage of healthy hepatocytes, but it doesn't seem to be following the expected results, which is.
Sorry, do you want to show that and let's take a look. Okay. That doesn't look too bad. Let's see, can you, again, if you could possibly resize so you could see the paper on one side, and there you go. Yeah, there you go.
Should I go back up to where the equation was?
Right.
Wherever the model was.
Oh, I see. Okay. So we did exactly the same thing. So if you'll notice, if you look in the figure 1, on the label of figure 1,
yes,
the amount of virus is very large compared to everything else. So to get them on the same axis, they divided the virus by 1,000.
Oh, okay.
So the best way to do this, I think, would be to plot, run your simulate once and plot just X and Y. So you'd add comma, square bracket, X, and you have to X. You actually have to do time, comma, X, comma, Y to make it work. Okay. And then now try that. So this looks like
that's
exactly that figure 1. And now you could just copy those 3 lines and paste them in underneath. And now instead of X, make it V. There you go. And now you can do that. There.
Could I like create an array of just a V where I just divide V by 1,000 and then
Sure.
use the plot array function?
Mhm.
Like I could create arrays for all of these.
Yeah, I mean, definitely, if I were going to do that in production mode, that's what I'd do. But for quick and dirty, I thought that was the fastest, probably the simplest one. But yes, that's fine. And if you wanted to plot the bottom plot, then you'd plot y divided by x plus y.
Yeah, I created a variable for that in my model.
Great, that works. That works great. Let's see, let's look at PRC again. You've got X over it. You've got that's the percentage uninfected.
Oh.
Y is the infected.
That's more like it.
So how are other people doing? Are people able to follow that or need help with that?
This looks like what I have.
I think it's probably better not to go on until everybody's caught up. So one thing, if people get stuck, I suppose, Kali, you could basically cut and paste your code into the chat, and people could work from there. But whatever, however you like. Or Brian and Mercedes, if you want to spend a little more time on it, that's fine.
Sure.
You tell me. You know, I shouldn't be. I don't want to be running this group. I want to just be there to help. If Brian, if you and Mercedes want to screen share, just say so and we can.
Yeah, I'd like to screen share. Because I've been rearranging my variables. I think I might have missed something. Okay, let's try this.
You're having trouble. Okay, great. Okay. So I think, again, I would recommend split screening so you have the paper on one side and the notebook on the other.
Mm-hmm.
So you don't have to switch back and forth between them. Because it looks to me like you have a little bit of a typo in your antibody, and it's much easier to see that if you have them both on the screen at the same time. I know when I have to switch back and forth, I mistype.
Let's try this.
Okay, so let's look at the equations. So I see you have R times X here. I don't see, so then you have Rx. Well, so remember what nothing goes to X does.
Mm-hmm.
What does it do?
Nothing goes to X.
Now, what is that equivalent to as an equation?
Basically just defines it, right?
No, but what is it if so if I have arrow goes to X, R times X, that's equivalent to a differential equation. What is that differential equation? But don't look at what you've got there. You typed it, so now I want you to write. You can use the annotate function. Write what the differential equation that represents is.
The annotate function?
Well, you go to Zoom, you can just type by hand, right?
Okay.
You're trying to represent this big equation here. You've typed this, so yeah. So what is this piece of it? And I know it's a pain to write by hand when you have a mouse. So I know it does. Okay. That works too. You can use the alpha. Okay, equals. Right. Okay, good. Okay, that's fine. So that term is there. And now you wanted to have X goes to nothing, and that would be this times this, right? That's what you're trying to write here. Is that right?
Right. Yeah.
Okay, that's fine. Looks like. Okay, and now you have X goes to Y, beta times V times X divided by X plus Y. That looks fine. And then Y decays A times Y. That looks all right. And then V is produced at rate gamma 1 times Y. That looks fine. And it decays at a rate mu. So this looks fine to me. Everything here looks fine. All right. And now let's see what happens when you run it. Okay. So the same issue that Kali was facing, which is that the amount of virus is much bigger than everything else. So it's better to you have to either split, what I suggested doing was plot them into two separate plots. So I would plot, why don't you just put in, there we go. And then remember the syntax is time, values. That, if you put that at the end. Would be X and Y.
What am I missing here? Is it time
Well,
and?
You need to have X and Y. You'd have to, no, time, comma, X. So you have to say, this is telling you what columns to output of your data set. Okay, so if you want to plot X and Y, you'd have to have time, comma, X, comma, Y. But we'll try that and see what happens. these have to be in quotation marks because they're the names of the variable. So X and Y. All right. So if you want to make it the virus, you make another one. This one would be V. Get rid of that. Well, you don't want to put the virus in the same one, so you would put it in the same. Right. You could get rid of the Y in the second one. All right, so it's certainly infecting your cells. It doesn't seem to be doing quite what you expect. So there are possibilities. Either there's potentially a typo in the equations, possible, or there's a possibility that the one of the parameter values isn't what you think it is.
Oh, there it is. It's because I had one of the equations not the same, different, a similar number. Like I had E3, and then it would just do that.
So that's something that is would in a procedural language, it would either throw an error or it will tell you. In a declarative language like this, later lines replace earlier lines. So if you have E3 twice, the second one will be what you use. Okay, good. Okay, great. So you've got this now going. Mercedes,
Nice.
how are you doing?
I have it running. I had
Okay.
what Brian had.
Okay. Okay, so everybody has that one. So let's see. So the next thing to do would be to get, I think, to get the, first you want to print out the value of R naught. So you maybe just print the value of R naught because you'll need it. And remember, R naught is equal to the rate of virus production times the infectivity. You may have called those by different variable names, divided by the rate of viral clearance times the rate of cell death. And so you probably want to print that out because when you change your parameters, you want to know how R naught changes. And then I would change R naught, and you could do that by changing any of the parameters. And so explore what happens when R naught is less than 1, when R naught is between 1 and 7, when R naught is between 7 and 9, between 9 and 10, and more than 10. Just do individual runs changing the value and see what happens. So why don't people take a few minutes to do that? So come back and let me know how it came out. Okay, I'll be right back. So whenever somebody has some results, or feel free to screen share and let's take a look.
So I was going back to what we were doing before. I had the plots that we were doing before, but I was trying to replicate the figure and having all the plots on one graph.
Okay.
And I tried to do that, but then I ran into an error, an index error.
Okay, let's take a look if you like. All other people are working. You want to screen share it?
Yeah. I got rid of the plotting code for now because I'm trying to solve this index error. But for the previous model, I was able to use this method where I created an array and copied the data from the simulation into that array and then used the plot array function.
Right.
But here it's complaining that only integers, slices, ellipses, etc., and integer or Boolean arrays are valid indices. So are we somehow maybe not getting integers here and it doesn't like that?
No, it's complaining about where is it complaining? Let's see.
Hold on, let me run it again. It's complaining here.
Oh. It's because so you got 2,000 elements. What it's having trouble with is the shape of it's complaining about not complaining about the right hand side of this, it's complaining about the left hand side of it. So you're doing a slice of 10,000. I don't know why it doesn't like the one, but it's that one that it's complaining about. This isn't quite how I would do it, so I'm trying to figure out why.
I suppose I actually don't have to do this method. I think for the previous model, we wanted each
Well, I think
on its own graph,
yeah.
so I decided to split them up. But
Right. I think you definitely will want to have something which is the data set, because you'll need it to do the remaining exercises. You'll need it.
Right.
So, but why don't you instead of putting it into an array like that, why don't you? So instead of creating that uninfected equals n pi zeros, why don't you just say that time equals s underscore HBV quote time to give you that column, and then x equals the second one. Like that. That should work. And so now you have the columns individually. And if you wanted to do something like plot them you could use a plot array now to plot the pairs if you like, or you could V stack them or H stack them if you wanted.
Mm-hmm.
Try that and see if that runs. If that crashes, then something else is going. But why don't you just execute that block and see if it gets through those 4 lines or 5 lines.
Still having the same complaint.
All right. So the problem is in that slice. Let's look back before and how we did it. Did we leave out a regular parenthesis or something like that? Can you just scroll up and see, look at the example you did before where you used that method? There's, okay. SHIV excluding at the time.
Are X and Y dedicated variables?
That's possible.
I suppose if they were, then wouldn't we have not been able to get past the loading and simulating the model?
Yeah, I'm a little. This has got to be one of these things which is obvious in retrospect. So let's just go scroll down. Let's see, scroll down our output. Underscore S HBV. Which one died? Time was all right.
Yeah.
But X was not. Oh, I know why. Because you did this twice. Yeah. The problem is here. You declared the names of the columns here, but then you ran it again without declaring the names.
Oh, why did I didn't even realize I had it there twice.
Time is always named, but X and Y aren't named columns unless you declare them.
Okay,
So if you take out those 2 lines, it should now work. Yeah.
Yeah. I don't know why that was there twice.
Yeah.
Okay,
So,
thank you.
Anybody else? How's it going for other people? Was anybody able to figure out what R naught is and try plotting, doing the same simulation with different values of R naught?
I think I found R naught, but then I got distracted by what you were helping Callie with. So
Okay. Well, if you've got it running, why don't you put it up on the screen and let's do it together.
okay, I only plugged in the equation of R naught equals beta times gamma 1 over m times mu, or a times mu. That's all I had. So.
But do you have the time series? Are you plotting the time series?
I don't think I just did the load and simulate. I didn't do any of the. I'll just share I'll share what I have.
Okay, that looks fine. So you printed R naught. R naught's 8. So the simplest way to make R naught smaller would be to change that gamma 1. And so the way you would change gamma 1, let's make it less than 1. So if we divide by 10, it's less than 1, right? So
sorry, I keep fighting back sneezes.
That's okay. So after the m equals t dot load a model string, You'll have to do it repeatedly because you're the first plot I think you don't need because it's duplicated. So I get rid of that first pair, that first triple of plots. Okay, so now you do T at load a model string. And now we're going to change the value of gamma 1. And there's an easy way to do that. You'd say m dot gamma 1, the way you did in the line below. And we'll do divided by equals, which is Python-y, 10.0.
Do you have to? I guess not. Never mind. We're not looking.
And you'll have to repeat that after that second m dot equals load because it resets. The load resets it. Okay, so now try that. Okay. So what happened? What happened to your infection?
It didn't happen.
Right. And what's your value of R?
0.8.
Right. So now let's make R just a little bit bigger than 1. So instead of dividing by 10, let's divide by 8. Well, it's just slowly starting to get infected, right? At the very end.
Yeah.
Okay. Let's divide by 5. Still pretty slow. Maybe we'd be better off. Let's start with instead of making our initial condition V1 equal to 1 e to the minus 6, which is really teeny, let's make it just 1. Try that. It's still pretty slow, but anyway, okay. R naught. Okay, so this is telling you how long things take. So if you wanted to see what really happened, we'd have to run it for longer. So you'd have to simulate for maybe 1600 instead of 800. There you go.
Okay.
And now let's make R naught bigger. Let's divide by 1. Go back where you started. So now it's oscillating and decaying. And I'm actually sort of surprised because I thought for R naught bigger than 7, it was supposed to, oh, this is the bifurcation. Okay, I thought it was supposed to oscillate. Let's divide by 0.5, make R naught bigger. Oops. Now everybody died. All your cells died. So maybe try 0.75. There you go. Now you got stable oscillation. So you saw if you make that gamma small, very small, you get no infection. For gamma a little bit bigger, the infection takes a very long time and you get a steady state. For gamma a little bigger than that, you get oscillations that die out. For gamma a little bigger than that, you get steady oscillations. And for gamma bigger than that, everybody dies. So I think it's worth everybody trying that on their own. Take a minute or two to try that on your own. Because that's really the critical thing in this paper is that. Let me know when you played with it enough that you feel comfortable going on a little bit. How's it coming? Was everybody able to get that to work?
Yeah, I saw the oscillating behavior in mine.
Right.
My graphs.
Okay, so the next thing to do is a little bit more let's see, what I'm trying to remember now what the order of the assignment was. The next one was so if we look at the paper, the paper has what we call a bifurcation diagram, where it plots x plus y, the long-term x plus y versus R naught. So what you'd do is you'd scan gamma 1 and plot and run the simulation for a long time and determine the final values of x and y and plot x plus y versus R naught. And to do that, you'll have to use the V stack that we used before. So if people can look that up, or if you need help with it, maybe it's worth trying to look it up yourself so that you can remember. But what you want to do is essentially run the simulation for long enough that the value stabilizes. And if it oscillates, you're going to have to think about what to do. And my recommendation, this is a little bit more work, but the way Kali, you've got it set up was not so bad for that, which is look at the tail end of your simulation when the oscillations have stabilized and find the maximum and minimum values. So I would run the simulation, add x and y together to get t, and then plot, and then look at the tail of that array, say the last 1,000 points of the array, and find the maximum and minimum. And then I'd plot R naught on the x-axis and the max and min of t on the y-axis. And you should be able to reproduce that plot that is in the paper. I'm trying to see if I can open that. I said everybody should have the paper open, and I don't have it open on my screen because I was looking at something else. Let's see. Okay, so you see this picture here, number 4. So this blue line there's a typo. So even published papers have typos. It should say x plus y, not x plus p plus y. There is no p. I don't know where they got the p from. It should be x plus y, the total number of hepatocytes. And then you have R naught on the x-axis. And to scan R naught, you have to scan one of the parameters that control R naught. The easiest one to scan is your gamma. The bigger gamma is, the bigger R naught is. And so I would do a loop where you scan gamma. You run the simulation to calculate. You don't need the time variable for that. Just get x and y. Add x and y together. Get the tail end of the distribution. And if people, like, let me get my, so if you have an array like this, if you do -1000 : nothing, that'll give you the tail end of the array.
I don't think we can see what you're typing.
Sorry, I just put it. Oh, sorry. Yeah. I put it in the chat, but I can also put it here. So what you can do, let me do an annotate also. So what you could do is say for gamma 1 in and pi, you need numpy loading, a range. Actually, it's a multiplier, right? Multiplier in numpy, a range 0, comma 2, 0.1. And then you'd say load. I'm not going to type all that load. And you'd say m dot gamma 1 divided equal times equal divided equals and pi multiplier. They're back. And then you could do calculate x. And you could say n pi dot max of x with colon, give you the maximum value and so on. You stack that. Okay, but let's take I'll get offline here and let's hear, and Josh, you could help maybe here too. Let's hear what people have. I think people on our end have made some progress. So let's hear where you are. And if we don't finish everything today, some of it can be homework and some of it could be done next week too. See how far we get. So who wants to talk about, who wants to speak first? Josh, how are things going? You're muted.
Oh, I'm sorry. I didn't know. Yeah, it's fine. I think Antonio can walk you through.
Okay, I'll let you drive, Josh. I'll turn it back to you, okay?
Okay, so Antonio is gonna explain. But Antonio, you might want to show at least the plots to show what the points is in the parameter space that we were exploring.
Okay, I can do that. So this is the implementation of the model without intervention. And so if we look at the paper, they have these kind of fields where they show that they do these calculations with the constants are included. So RS is some combination of the wild type constants. RR is another formulation of the resistant type constants. And so we see that in this space, and this is RS equal to about 1, slightly over 1. In this space, they coexist together. In this space, we see that the resistant strain actually outcompetes and eliminates the wild type strain. And in this space, we see that neither strain succeeds and they both die out. So the first thing we did was we kind of, we used the KR value, which is the mutant infection rate. And so we changed that around to kind of change the values of the RRs to get into those different spaces. And so in this first space, we did it just with the parameters given to us in the model, and it shows a coexistence between the wild type and the mutated type. And these are the R values that you can look at in that space. Then we ran it again and adjusted the R values so that the mutated will outcompete the wild. And here we see that the wild type strain does indeed die off as the mutated strain thrives. And your total viral load is just your resistant strain.
Can
And
you
then we
show?
did the opposite way.
Can you show how we moved in the parameter space in this case? We moved from RS3.
Yeah, so we were about here initially in the parameter space, and so we multiplied that KR value. So we're way on the right of that graph. I think we had an RR of about 16, and then we divided both by a magnitude of 10, and we ended up way down here in the final one, where the wild type dies out, mutated dies out, and we see that the total viral load also dies out. So then we moved on to applying the intervention, which was the addition of this epsilon value. And we did a code monkey way of scanning through the efficacy of that epsilon. So we started with an epsilon value of 5, which basically says that the reverse transcriptase inhibitors are 50% effective. And so we see that, initially with the efficacy of 50%, that it doesn't really do much to either strain. And then if we look in the paper, that kind of concurs with these, the graphs that the researchers give us here, where they plot these as they scan through this efficacy against the wild type. And so at 0.5, we see that the wild type shouldn't die out by the end of it. This neither should die out, and we should have a total viral load. So then we kind of we bumped it up to 0.7, and we see that with the efficacy of the RTI being 70%, the wild type dies out, but the mutated strain is able to continue operating. And then that concurs again. We're in this kind of pocket here where we should see that the drug-resistant virions survive with an ES of 0.7. Additionally here, while here we see that the wild type all dies out. And then lastly, we have bumped it up to 90%. We see that the wild type dies out. We see that the mutated also dies out, and it forces clearance of the entire viral load.
Great. So now, in principle, you could do a parameter scan for that, and then save the final values and stack them, and you could reproduce those plots.
Right.
So that would sort of be the next, because we're going to run out of time, but that's the next exercise. So maybe that's a homework problem or something we could return to next time. But that's great. So you're basically able to reproduce the whole paper.
Yep.
Which is, it will certainly work, I'm sure, but being able to reproduce a major paper in a few hours is not bad.
40 minutes.
Oh, pretty impressive. So that's great. So how about on our end? Do people want to show where we got? Because we also got through quite a bit. We didn't get to the phase diagram yet, but we got through quite a bit. Does somebody want to present for the Mercedes? Do you want to show what you had? Or Brian? Or Jeffrey? And any of you? Somebody speak up. Kali presented last time, so I'd like somebody who didn't present in the previous round to be the presenter this time.
Yeah, I can share my screen.
I'm not trying to discriminate against you, Kali, but just.
Do you want the paper up at the same time or just the?
Yeah, it wouldn't hurt if you could put them both
Okay.
up.
I'll do both. So here was the paper that we were at, going with the HPV. And then we got through the point of looking at different levels of R0, so by adjusting gamma. And basically we found that as you get to the right level of R0, it gets to the oscillations. So you get infected and or not infected, but you don't die out and you get the constant oscillations. when you had too little of an R naught, nothing ever happened, or it took too long to even see what was going on. After we ran the smallest R naught, we adjusted the initial number of we adjusted the initial V1. We started out with it really, really small, and that caused a lot of delays. And then if you made the R naught too large, it got to the point where everybody died. So there was no way to recover it or get the constant oscillations. So
I think you had one case where you got decaying oscillations too.
yes, that was I don't remember which case that was.
I think that would be for R naught slightly smaller than the one where you had the steady oscillations.
Yeah, I think we did a gamma even in between the 0.5 and 0.75.
So again, the thing to do next would be to try to sweep gamma, and then keep track. The thing you're going to have to do with these oscillations, which makes the case this one a little bit more complicated than the other one, is that if it's oscillating, you have to keep track of both the top and the bottom of the oscillation. And so you can't just ask what's the value at the end of the simulation, because that'll depend on where you are in the oscillation. So instead, what you have to do is you have to save the time series, and then you can use the NumPy max and min functions to determine what the maximum value and minimum value are. And if you scroll through the paper, if you scroll down in the paper a little bit, the next thing I would try to do is, if there's a nice figure, keep going. Next one, right there, stop. Is this figure here. So the blue line is X and plus Y. The bottom line is R naught. If you reach a steady state, there's only one value of T. If it's oscillating, the oscillation goes up and down between the upper blue line and the lower blue line. And so if you find the maximum value of this oscillation, that'll give you the upper blue line. You find the minimum value, it'll give you the lower blue line. And so the next step is to do that plot. And so one has to look up from class notes how to do the VStack to save and plot those. But I think that everybody probably can figure that one out. If not, we can ask. Okay. So I think that we're out of time. Is there anything people want to discuss before we break tonight? And Josh, maybe you can write up a little homework to help people go through this. But I think I'm quite impressed that everybody was able to get the time series running and change the R naught parameter and see what happens. And so I understand it's non-trivial to replicate papers, but the only way to learn how to do it, I think, is practice. So I hope this was at least somewhat useful for people. And it looked to me like everybody was able to do it, which is good. So is that anything else, Josh? I'll turn it back to you to wrap up.
Right. Yeah, I'll think a little bit about what to assign for the homework. Whether we want to have you guys just do the parameter scan by yourself, or we can spend a little bit of time next week working on that. But we also want to move on to different topics. So I'll discuss that with James. But you all basically have got what they did in the paper, except that instead of exploring the points manually, what you want to do is to wrap that so you explore them systematically and record what the values at the end of the simulation.
And I think it really would be nice to put in some text boxes to put in the description of the paper. Because
Right.
both of these are things that would be really nice to have as nanoHUB apps.
Right. And in Antonio's case, for example, if you can include an image and then point the image of the parameter space, or in both actually, in both, and then mark which one, which point in the parameter space you're exploring, then it becomes easier in terms of walking through the notebook.
And I think, Josh, maybe next time you could show them how to use a slider to change, say, gamma,
Right.
and have it replot.
Right.
Because that's the one thing that they'd need for a online exercise, packaged exercise.
Oh yeah, then we can do, right. Then we can, yeah. Okay, I'll do that. I'll do that next class.
Because it's not difficult, but it does take it's not something we can do in -5 minutes.
Right.
It takes 15 minutes to show how to do it. Right. But once you have that, you could actually make this a very good educational exercise for people. Mm-hmm. I think it's worth putting up both of these online. I think they're both really good demonstrations. Okay. Anything else, Josh? Okay, so I'll let you wrap up the evening.
Okay, good. Then thank you, everyone, and we'll see you on Tuesday. We'll post the homework later today or tomorrow morning.
And I guess I said I'd let Josh wrap up, and I said, please email and so we can schedule meetings on your projects.
Oh yeah, please do that. Yes, absolutely.
But I hope going through this helps you think about the project. Because the process we're going through here is basically what you have to do for your projects too.
Thank you.
Okay, thank you, everybody. Good night. Appreciate it. Get some rest. Get something to eat and get some rest. Okay.